Hello everyone, my name is Ashley Candeletti, and this is my Aquatic Microcosm project for American Public University's General Ecology course. According to the Encyclopedia of Ecology, microcosms are defined as simplified physical models of an ecosystem that enable controlled experiments to be conducted in the laboratory. When taking on the microcosm experiment, I was not too sure what I would expect. I was unsure of what a microcosm really was and how it helped me understand the natural world. After much research, I realized that using microcosms creates a controlled way for ecologists to study the environment. They're able to use these to control certain factors, abiotic and biotic, to determine the possible outcomes for our future. Once I learned out what a microcosm was and how it affects the field of ecology, I was hoping to learn how the organisms in this particular microcosm would survive in an environment where all abiotic factors were controlled. For my microcosm experiment, I had two hypotheses. My first hypothesis was, if the aquatic microcosm is to thrive, both the autotrophs and the heterotrophs will be alive at the end of the project. My next hypothesis was, if the aquatic microcosm is to be successful, the water will continue to be clear. To create my microcosm, I took samples from Lake Audubon in Western Virginia and placed them into a 32-ounce mason jar. I filled the jar about 5 centimeters high with the mud, which consisted of sand, silt, clay, and rocks. After this, I filled the jar with water and let the material settle. Once the water and mud had settled, I had added a leafy green plant called pondweed and a snail into the microcosm. I monitored the microcosm for 7 weeks, in which the light and temperature of the environment was controlled. There was about 12 to 14 hours each day of natural light, and the temperature was steady at 70 degrees Fahrenheit. Over the 7 weeks of this project, I was able to observe how the microcosm thrived. Originally, the plant started out very green, and as time went on, it became more brownish, showing that there was less photosynthesis in the next few weeks, but finally it returned to more greenish color in the last week. The water also started out slightly yellow and cloudy, but became increasingly clear over the time. The snail gained more algae on its shell and came out more in the last week. In the 3rd and 4th week, a worm appeared, but I was not able to see it again after that 4th week. The mud continued to be a dark brown color, but there were more and more cracks seen in the soil from the increasing amount of roots as well as the worm. This is a timeline of my experiment. I took a picture on every Sunday afternoon, and I was able to use this to track and record my observations that I spoke about on the previous slide. Both of my hypotheses for this experiment were accepted. The plant and animal both survived, the plant stayed green to indicate photosynthesis, and the snail continued to live, moving to different places periodically within the microcosm. The water also stayed clear through the end of the experiment. Originally, the water was quite murky and yellow, but it cleared up after the mud settled, and the snail and plant were able to keep the water clear. To improve my project, I would create 3 control environments, one with no organisms other than what is in the water, one with only an autotroph, and one with only a heterotroph. Some hypotheses that I would also like to test in the future would be how light affects a microcosm, how temperature would affect a microcosm, and how a species from a different location would survive in the same microcosm. To relate this project back to class, we can look at 3 different topics from our lectures and readings. The first is species interaction. Both the pondweed and snail survived due to the autotroph and heterotroph having relationship. Their biological processes of photosynthesis and cellular respiration occurred to allow oxygen and carbon dioxide exchange. The next is adaptation to the environment. Both the pondweed and panhandle pebble snail had to adapt to different conditions. The conditions were constant, but the outside temperature was cooler, and there was more direct light on the microcosm within the experiment than was seen in their natural environment. This was seen through both organisms since they had to become accustomed to the new environment. The last one is climate. The climate was regulated in this environment. I tested how the plant and animal would survive within the climate when it was steady and not changing. In the field, microcosms can be used by ecologists to study the changing climate around us. Since these are controlled environments, they are able to see how certain factors affect species, populations, and communities. They're able to find and understand environmental concerns and test possible solutions to environmental issues.